Good morning, good evening, everyone. Welcome to the 84th Emerging Growth Conference, day 2 of our 2-day virtual investor conference. I'm Anna Berry. A few notes. Today we're running until about 4 Eastern. When we switch to the next company, you'll see a black screen for a moment. Don't go anywhere. That's just us moving over to the next presenter. If you do experience downtime, refresh your browser. And our platform does work best on Google Chrome. So if you're watching from an Apple device, you have to hit the play button to start the session. Now, during each company's presentation, you can submit questions through the webcast module, and we will attempt to address as many of these at the end of the presentation. All of our conferences, they're uploaded to the Emerging Growth Conference YouTube channel, so please subscribe, youtube.com/Emerging Growth Conference. One last note, after today's event, you'll be redirected to the registration page for the next conference, so stay on or come back to reserve your spot early. Happy to begin with Argenica Therapeutics Limited, trades on the ASX under the symbol AGN. It is developing novel neuroprotective therapeutics to reduce brain damage after stroke and other devastating brain injuries. Happy to welcome CEO and Managing Director, Dr. Liz Dallimore. Welcome, Doctor, to the conference today. We're happy to have you. The floor is yours.
Thank you so much, Anna, and thank you for the opportunity. I think probably the Argenica story is quite new to a lot of the listeners out there and a lot of the viewers. So, looking forward to talking through a bit of background to the company and some exciting catalysts that we have coming up for the company. So just quickly, a disclaimer in terms of any forward-looking statements within this presentation. So Argenica as a company, and as you said, we are developing what's known as neuroprotective therapies. So these are drugs that are able to protect the brain following brain injuries, such as you mentioned, stroke. Really our mission as a company is to commercialise these treatments, so take them through the clinical development pathway and really be one of the first companies that can revolutionise the standard of care for patients that are suffering really catastrophic brain injuries following stroke, following traumatic brain injury, and we're also delving into neurodegeneration as well. So our lead drug candidate is known as ARG-007, and it's what we call a cationic polyarginine peptide. So this just means it is a drug that's made up of many arginines, and really it is this arginine content that confers its incredible neuroprotection that we see in particular in our preclinical studies. What's really unique and a really key differentiator of our drug is that it works on multiple mechanisms of action. In neurology, this is really important because neurological conditions are really, convoluted. So there's a lot of different things that are going on. And so if you have a drug that can block a number of those different adverse pathways that we see in these neurological conditions, then you're going to have greater efficacy and a better drug. We have a really strong IP position within the company. The drug itself is actually spun out of a research institute here in Perth in Western Australia and has over a decade of research that's come from that research institute and into the company. The company does now own all that IP, and we're free from any encumbrances associated with the transfer of that IP. As I said, significant preclinical efficacy data on this lead drug candidate ARG-007, which is covered in over 25 peer-reviewed publications looking at both the safety of the drug, the efficacy of the drug in a number of different stroke models, and I'll come to that a little bit later, as well as traumatic brain injury and a number of other indications. We've also tested the drug in healthy volunteers in what's known as a phase 1 clinical trial, and we saw no adverse events, in particular in that top dose, which is a dose that we've taken now into a phase 2 trial in stroke patients. So far the drug is looking really good. We've got fantastic preclinical data and a drug that has a fantastic safety profile, which is a really big plus in drug development. So as I mentioned, we are looking at stroke as an indication. This indication is coming really to the pointy end of our phase 2 clinical trial, and I'll talk about that in a bit more detail later. We're also looking at severe traumatic brain injury, and we've got some really encouraging preclinical data in a number of different traumatic brain injury models across both rodent and ferret models. So we're looking to move that indication now straight into a phase 2 clinical trial. We're also working a lot on a rare paediatric condition known as hypoxic ischemic encephalopathy. We're actually working really closely with a group out of the US, a consortia, where we're looking to work out the best way to run clinical trials in this, what is an absolutely devastating condition that affects newborn babies. We also have a program of work looking at the more mild and moderate TBI. So this would be things like concussion. And we also are starting to delve into the neurodegeneration space as well. So where do we sit at the moment? So I probably don't need to let the audience know that these indications, stroke and TBI alone, are huge addressable markets. So the treatment markets for those alone, you can see there, $12 billion and $18.6 billion respectively. What I've got on the right-hand side of the slide is, and showing the audience really what our drug is able to do in the ability to reduce what we see, the brain injury that we see in these preclinical animal models. So the thing that gave us real confidence in stroke was a non-human primate study where we showed that our drug was able to reduce brain injury up to 70%, which is hugely significant. Similar data that we're seeing now in our traumatic brain injury models as well, which is really exciting and really encouraging. So it really, de-risking the drug asset using these preclinical models is really important as we move into clinical trials in actual human patients. So just to touch quickly on the company metrics. So as Anna said at the start, we are listed on the Australian Stock Exchange, the ASX, with the symbol AGN. As of the end of the March quarter, we had a little under 13 million, that's Australian dollar, cash in the bank. That's plenty of cash to get us right to the end of our phase 2 clinical trial and the data readout. We're sitting a little under 100 mil Aussie dollar market cap on the ASX, which is a reasonably sort of small market cap for the stage of development of where we are. So plenty more upside, definitely within the company. And we've been really successful in attracting that non-dilutive funding, government grants to help us progress these indications. What's not included there is a recent $1.5 million grant that we've just received to progress the stroke indication. So I just want to give the viewers a little bit of an update on ischemic stroke and really why Argenica is focused on this indication specifically. So what people might not understand is just how prevalent stroke is and how more prevalent it is actually becoming. So in the US, we know now that every 45 seconds, someone suffers an ischemic stroke. So this is a stroke caused by a blood clot in a blood vessel in the brain. Ischemic strokes are the most common type of strokes. They make up around 85% of all strokes. The other type of strokes are what we call hemorrhagic strokes, and this is caused by a bleed on the brain. Argenica is going after that bigger indication, those ischemic strokes, so those clot-causing strokes. So of these people that are having these ischemic strokes every 45 seconds, only around 10% of them will make a full recovery due to the extent of brain damage. And what we know is that as soon as that clot gets blocked in a blood vessel, brain cells start dying almost immediately. And you get this proliferation or cascading out of brain cell death that we see in stroke. And around 1.9 million brain cells are dying every minute. And what we don't have is a drug that can halt that, stop that cascading out of brain cell death that we see in stroke. We need a drug that can buy these patients time to get the treatments that they actually need. So really what Argenica is going after is to be a first-in-class drug, the first neuroprotective drug on the market that is able to protect brain cells in these stroke patients. So what does that look like in terms of the actual opportunity and the size of that market? So we compare this to a drug that is currently used in stroke patients, which targets the actual clot and aims to dissolve the clot. This drug is known as a lysis drug, TPA or TNK, a thrombolytic type of drug, and really it's designed to essentially munch up that clot. So we know the cost of that drug, so that's around $10,000 per patient. And the total market size of that is projected to be around $3.8 billion in 2030. So only of the patients that receive, these ischemic stroke patients that receive these thrombolytic drugs, only around 9% of them can actually receive it because it's quite restrictive in terms of the types of patients that are actually eligible for that drug. So if there's only around 9% of patients that can receive it, and it's a $3.8 billion US market, the patient population Argenica is going after is almost 4 times that. So our market size and our opportunity is closer to the $10 billion range. So absolutely huge opportunity for Argenica. We are one of the leading global companies. We're the furthest progressed clinical global companies developing a neuroprotective drug. So it's a really exciting time for the company, and I hope everyone listening out there gets as excited as I do about not only the market opportunity here in stroke, but also the potential to be an absolutely life-changing drug for these patients. So what do we have today in stroke? So as I mentioned before, we have done non-human primate studies. This comes off the back of a huge amount of data we have in rodent studies, in stroke models. But what really gave us the conviction to actually take this drug into clinical trials in stroke was the fact that our drug could reduce the total amount of brain injury following an ischemic stroke by 70%. Which is absolutely huge. So, and we noticed that what we saw in those studies was the ability for the drug to reduce that brain injury by 70% at 24 hours post-injury, but it also remained out to 28 days. So it wasn't a transient effect of this drug. The effect of this drug was long-lasting, which is really important. We also know that our drug is safe to administer regardless of the type of stroke a patient is having. So the problem with the thrombolytic drugs that I was talking about before is that if they were to be given to a hemorrhagic stroke patient, so a patient that has a bleed on the brain, those patients would bleed out. So it's really important with thrombolytic drugs that the patient is diagnosed by imaging to determine the type of stroke that they have. For our drug, it doesn't matter. So we would be able to eventually, hopefully, give our drug in the ambulance to anyone that is suspected of having an ischemic stroke, regardless of if they're having a hemorrhagic stroke. From a clinical uptake point of view, that's a really important point. We also know that our drug can be administered with the thrombolytic drug, the clot-dissolving drug. Again, from a clinical drug development point of view, it's really important to show that any experimental drug, any new drug will not impact a drug that is already standard of care. So we've shown that as well in a number of studies. And as I mentioned, we've conducted our phase 1 clinical trial already. So we've shown that the drug is safe in healthy humans. So that gives us a kind of tick of approval in terms of the safety profile of the drug. So I'll just talk a little bit around our phase 2 clinical trial. This clinical trial we've completed dosing of all the patients in this trial. And so we really are coming essentially to the end of the trial and the data readout, which is really exciting. So essentially in this trial, what we're really showing as the primary endpoint is safety. So in any neurology drug development, it's really the biggest hurdle from a regulatory point of view is to show that your drug does no harm to these patients. Once we show that it does no harm, that's the first hurdle to get over. And then we look at to what extent is the drug actually reducing that level of brain injury, what we call the infarct volume. So how much is it reducing that? So the trial design for this this phase 2 clinical trial is we've only focused on patients that have a clot in a large vessel. So these are the most severe types of strokes and the patients that need essentially a neuroprotective drug the most. Around 40% of all ischemic strokes are large vessel occlusion strokes. So a patient comes into hospital, we still want to screen these patients, we still want to do the imaging of these patients to confirm that they've had a large vessel occlusion. So that's usually done via MRI or non-contrast CT within the emergency department. Once we know that that patient has a large vessel occlusion and that they are eligible for our trial, they receive a 10-minute IV infusion of either our drug or a saline placebo. And really important to note that the entire study is blinded. So no one involved in the study knows whether the patient has received our drug or the placebo drug. One of the other key inclusion criteria is the patient then goes in to receive a procedure known as a endovascular thrombectomy. So this is essentially where a neurointerventional radiologist will put a little shunt either up into the patient's groin or into the radial artery, up into the brain, and actually aspirate out or suck out the clot to remove it ideally in one piece. I'll talk a little bit about why that's really important. It's a really important distinction around our clinical trial design and why we've designed it that way. So just a little bit more then on the timelines for the trial. So the trial recruitment actually went really quickly. We initially had anticipated that dosing would take around 2 years to dose 92 patients across 10 hospital sites in Australia. We were actually able to bring those timelines in 12 months. So we completed dosing in 12 months, which gives a really fantastic indication of just how easy our drug is to administer to these types of patients in an emergency setting. There was a few questions, especially from a number of institutional investors who were challenging me at the beginning of the trial to say, you're not going to, there's no way you're going to be able to recruit 92 patients in 2 years. It's going to take so long. You're in an emergency department setting. It's going to be really challenging. So fantastic, I guess, to prove them wrong and to show that in these types of trials with our drug and the ease in which our drug can be administered, we were able to bring that recruitment in 12 months. So as I said, safety and tolerability of the drug really is that primary endpoint, but we do have that preliminary efficacy. So I think the market is really hanging out to see what the efficacy does look like. We have had a data safety monitoring board monitoring the essentially monitoring how sort of safe the drug is looking. They're all blinded, so they don't know if patients receive the placebo or our drug, but they haven't seen any essentially serious adverse events that could be possibly related to drug administration. So that gives us a really good sense that the drug is looking really safe. And we anticipate that top line data will be out in Q3. So we're thinking it'll come out. We'll announce it to the market in early September, which isn't that far away. So I would encourage everyone to get on board now before we hopefully get what is some fantastic data out of this trial. So I'll just quickly talk about then what that secondary endpoint efficacy infarct volume reduction is that we're looking for in this trial. And the reason that we're including thrombectomy patients, which is that clot aspiration. So we are delivering our drug what could be reasonably late post the actual stroke. So to give our drug a second chance of working, a second chance to see efficacy, we're only including patients that have this thrombectomy, because what we know in a large majority of these patients, even though the clot is getting removed through this thrombectomy procedure, they actually have a secondary injury known as a reperfusion injury. So we're still getting this infarct volume. So what we're hoping, knowing that our drug is already in that patient by the time they're going in for the thrombectomy procedure, our drug should be having a second chance to work by protecting against that secondary brain injury, that reperfusion injury. So you can see it here on the slide. The yellow is this area that we call the penumbra. It's a really vulnerable brain tissue that could post-thrombectomy turn into this white area here, which is essentially dead brain tissue. So what we're hoping our drug will do is in that yellow area, it will protect that brain from dying. So when we compare the two, essentially we'll be looking at the placebo, where that penumbra will turn into that dead brain tissue, and hopefully our drug prevents that. And we'll look at the two, and we'll look at the difference in that infarct volume reduction. So I've been spending a lot of time with investors around, well, what does success then look like in terms of that efficacy? How much brain do we actually need to save? How much infarct volume reduction do we need to see? And so there's a lot of clinical papers that have been published really asking this question. So we know what the minimal amount that is clinically important to patients, so what that difference is, and it's only a 1.6% reduction in that infarct volume, which is really small. And then it's kind of linear. So essentially, if you can reduce 5% of brain injury, you get around 5 patients per every 100 patients that you treat having a better functional outcome, so reduced disability. If you reduce infarct by 11.5%, you get 10 patients out of every 100 having a significantly reduced disability. 17% leads to 15 more patients, and so on. So it is reasonably linear. So we essentially, to show success in this trial, we don't have a huge hurdle. So safety is a big hurdle, and hopefully we get over that. But in terms of the amount of infarct volume reduction that we're trying to see in this trial, it's actually a really, really small amount. We are sort of aiming for that sort of 5 to 10% would be absolutely ideal. That's the amount that clinicians sort of say is a real game changer in terms of that neuroprotective therapy. So as I said, this infarct volume data will be out in early September, and we'll be able to look at the difference between the placebo and the treatment arm at that point to see, well, how much of that infarct volume is the drug actually reducing. So very exciting times. So I have a few more minutes just to quickly go through the other indications that I mentioned at the beginning of the talk. In particular, I wanted to talk a little bit around traumatic brain injury. So there are no neuroprotective drugs available for patients that suffer a moderate to severe TBI. Which is obviously a horrendously devastating condition and does seem to be getting more and more prevalent as well. So we've done a lot of work in animal studies, as I mentioned, both rodent and ferrets, and we consistently see the ability for our drug to significantly reduce what we just see as these axonal injury markers. So when someone suffers a traumatic brain injury, you get an aggregation of proteins within the brain cells that causes the injury to those brain cells. So what we do in these studies is we quantify the level of those proteins that are essentially aggregating. And this gives us an indication of how severe the traumatic brain injury impact is to those brain cells. So in this is the first rat study that we did, where we showed the ability to reduce neurofilament heavy protein and amyloid precursor protein right back down to normal levels. We repeated this study with more animals, and again, we consistently showed the ability to reduce these proteins, as well as some key inflammatory markers. So here it's IBA1. So the ability, again, to reduce these inflammatory markers. So we did the rat studies. That all looked really encouraging. But really what we wanted to do was to look at a larger animal study with a brain anatomy that more closely reflects the human brain anatomy. For traumatic brain injury, it's really important to test these drugs in models where the anatomy has what we call the sulci and gyra, those convolutions in the brain that can change the way that forces are distributed in the brain. So really needed to mimic the way that the forces would be distributed in a human brain if we were going to take this drug into a clinical trial into humans. So again, we saw hugely encouraging data from the traumatic brain injury studies in ferret. We've actually just repeated this ferret study out to a longer period of time. So it's really similar to stroke in, the idea would be that the patients would come into the emergency department, they would have a 10-minute IV infusion of our drug, and then we would look at the impact of our drug versus a placebo on brain injury. And we would be collecting what we call these biomarkers, so understanding how severe the aggregation of these key proteins that are the hallmarks of brain injury are occurring in these patients. So the latest study we did actually looked out to 14 days post-injury. So even though it's just a 10-minute IV infusion, actually goes the effect of the drug is lasting out to 14 days, which is really critical in this indication. And as I briefly mentioned, we are collecting a significant amount of preclinical data in that rare paediatric condition known as hypoxic ischemic encephalopathy, or HIE. And again, in these rodent models, we're moving now into larger animal studies in piglets and lambs as well. So we've got orphan drug designation from the FDA around this indication as well. So that provides us with some benefit should we move this into the clinic. So I'll quickly just wrap up on, what I really see as these key investment highlights for Argenica. So we are solving these huge unmet medical needs. There are no neuroprotective drugs on the market for stroke, for traumatic brain injury, for HIE. So, it's there for the taking, and we are one of the furthest progressed global companies in neuroprotective drugs. We have a huge body of evidence of our drug ARG-007 in that preclinical data package, which gives us the confidence as we do progress into these clinical trials. We've got really exciting near-term catalysts coming up, in particular around our phase 2 clinical trial readout, and we're having some really robust partnering discussions, which will really ramp up post that phase 2 data readout. So I'll leave it there, and thank you everyone for your time.
Wonderful. Thank you, Liz. We do have some questions, lots of questions for you. Let's see how many we can get in before we need to end. A few questions, a little bit about the competition. Emmy asks, who is your biggest competition, and what's the current default solution or treatment to what you're addressing?
Yeah, so I'll address the first part of that first in terms of the default. So essentially what these stroke patients, the only treatment really available is focused on removing the clot. So either through the thrombolytic drug, which dissolves the clot, or through that thrombectomy procedure, which sucks out the clot. So really what a neuroprotective drug is trying to do is trying to be an adjunct therapy to that. So it works in combination with both those treatment options to give the brain further protection. As I said, there are these reperfusion injuries once you do remove the clot, so the neuroprotective drugs work on that. In terms of our competition, so there's a couple of key companies, a Canadian company called Nono Inc. They had a couple of failed phase 3 trials. They've redesigned their drug to go back and test it again in phase 2 clinical trials. Their drug works quite a similar way to us. We've actually published quite a number of papers comparing our drug to their drug, and our drug always outperforms in terms of efficacy. And another company that we follow closely called Aptar Targets, which is a Spanish company. So they completed their phase 2 clinical trial actually a couple of years ago, and they showed some really good results in terms of infarct volume reduction. Their drug works very differently to ours. It really targets that later stage inflammation as opposed to the way that our drug works, which really stops the acute calcium influx. So potentially the drugs could work synergistically. Aptar Targets have actually just been, their drug has just been licensed by Merck. So, which is great for us because it shows that Big Pharma is really interested in this space and they're willing to do deals post phase 2, which is exciting.
Perfect. And last question real quick. Jim asks, is neuroprotective therapy your technology? Is that the IP? And if so, is it protected?
Yeah, so we've got extensive IP coverage. So we've got both composition use patents, and it's really around the structure of the peptide and the number of amino acids within that. It's a really broad protection. So any peptide drug that has at least 12 to 32 arginines in them, because it really is the arginine content that confers the neuroprotection. Any peptide drug ever developed in the world that has that will infringe on our patent. So it's a pretty comprehensive IP position.
Fantastic. Well, Liz, thank you so much for your time, and for everyone interested, it's Argenica Therapeutics on the ASX, ticker AGN. We appreciate you being here, important work you're doing, and please, we look forward to having you back on the conference real soon.
Thanks, Anna.
All right, everyone, we'll be right back.